la première présentation qui va nous être faite par Liliana SOFRONIC, qui va nous parler des aspects plutôt bénéfiques des parasites en tant qu'immunomodulateurs vis-à-vis d'une réponse immune trop forte. Liliana SOFRONIC est un médecin, immunologiste. Elle a travaillé très longtemps à l'Inep, à Belgrade. L'Inep, qui est l'Institut de recherche, c'est un peu l'équivalent du CEA pour la Serbie. Et Liliana a travaillé sous la direction d'un vétérinaire spécialiste de parasites, spécialiste d'immunologie et spécialiste de trichine, qui s'appelait Costa Kuperlovic.
Good afternoon. I like to express my gratitude to Professor Jean Bocamé and esteemed academicians for inviting me to participate in this meeting. And I really feel sorry that due to sudden illness, I'm not able to participate on site. So I devoted this slide to my mentor, Professor Kosta Čuperlović, a renowned immunoparasitologist who contributed a lot in the field of trichinella and trichinellosis in Serbia. And he participated in implementation of artificial digestion in all slaughterhouses in Serbia during the 80s in previous century. And he also was a member of an important committee that was a joint committee in between Ministry of Health and Ministry of Agriculture, with the main task to react under in case of re-emergency of trichinellosis and to propose measures for better control of spreading this infection to humans. I'm very proud of this slide because due to his efforts and later on efforts of my group, we were able to follow the presence of trichinella infection in domestic pigs and in human for 60 years. But as you can see, from this slide, in Serbia, parasites of Trichinella genus were detected initially in pig carcasses in 1918, while the first cases of human infection were recorded in 1923. The improvement in swine production practices had cut the prevalence of Trichinella infection in swine from 0.11 to in 1918 to less than 0.009 during the 80s. And Professor Čuperlović published a lot of papers where you can find the data for previous century. At the end of previous century, we had a big re-emergence of trichinellosis during the political upheavals. We had civil wars and sanctions, and we needed a quite long period of time to overcome that problem. And as you can see today, we are keeping under quite good control the level of infection among domestic pigs, and we have just a small number of cases each year. Only during the 2021, we didn't have any case of trichinellosis. So you can see that trichinella provokes a lot of different kinds of problems in my country. And if you like to read the papers, there is one I published regarding the first decade of this millennium, and the second one where I also participated, published by Sasha Vasilev, is full of details regarding presence of Trichinella infection in the last decade of this millennium. Since you could see from the previous slide that in this millennium we had around 3,000 cases of trichinellosis with only 3 deaths, and the main cause of the infection was Trichinella spiralis, although we have Trichinella britovi in wild boars. So there were some outbreaks that have been introduced by Trichinella britovi. But what we as immunologists have been very much interested in, we are providing first help in detection of Trichinella infection presence by developing at Inep in vitro diagnostic tools, indirect immunofluorescence, ELISA, and Western blot. But besides those kind of work, we were able to see that most of patients recovered completely and had no symptoms of trichinellosis, but some of patients in Serbia declared that they have noticed the improvement in their health condition, meaning there was absence of allergy symptoms they previously had, asthma, or relapses of labial herpes simplex infection. All those data indicated to us that trichinella can somehow help to healthy status of the patient and to better fight with other threats. Next slide, please. I will not bore you with a lot of data that we immunologists like to talk about. This is a complex network and communication in between parasite and different cells on the level of small intestinum. But when the parasite develops in muscle, then you can see first inflammation and then disappearance of inflammatory response. And I like to estimate that each stage of Trichinella first triggers an inflammatory response of Th1 type on the site of tissue penetration by parasite, but the parasite successfully reversed that response to Th2 type. Next slide, please. Those are nice slides prepared by Dixon Despomier, explaining how this amazing parasite is able to create a completely new type of cell in the host body, which name is nurse cell. You probably know that, and that nurse cell is completely free of muscle-specific proteins, and which had the sole purpose to serve the needs of the parasite. Next slide, please. And those members of Trichinella species are able to form the capsule. Capsule is a semi-permeable membrane, which allows nutrition from the host and metabolic products of parasite to pass through, and do not allow effector cells, immunocompetent cells, and specific antibodies to hurt the parasite. So during the evolution, parasite form this immunoprivileged place in the host body and take the host's immune response under its wing as long as it is alive. Next slide, please. We asked ourselves the same question as famous immunoparasitologists Bill and Margaret Harnett put. Can parasitic worms cure the modern world ills? So you probably know about existence of hygiene hypothesis telling that there is inverse correlation in between presence of parasitic infections and life-threatening autoimmune diseases. And we immunoparasitologists like to find out how parasite can help in curing those kind of disease. So keeping in mind that we are focused to Trichinella, we are studying what immune cells are involved, what are signaling pathways in those cells, what kind of cytokines they produce, what extracellular vesicles duty is in communication with the host, and we are looking is there any kind of help of application of nanoparticles and advanced therapeutic approach to chronic inflammatory and autoimmune diseases. Next slide, please. So first, we were able to show, we were first to show that Trichinella spiralis infection is able to provoke amelioration of the clinical course of EAE. It is experimental autoimmune encephalomyelitis that is used in as animal model for human disease, multiple sclerosis. That is a devastating autoimmune disease leading to progressive deterioration of neurological function. And we found that with 500 muscle larvae that we used for infection, we could successfully modulate those disease since we lowered all the signs of the infection comparing to control group where disease only was introduced. So duration of illness and infiltration of central nervous system were significantly reduced. Next slide, please. So we wanted to understand what are the mechanisms that are involved in this immunomodulation, and we were first who discovered that Trichinella spiralis influence maturation of antigen-presenting dendritic cells to keep that cells in semi-mature stations. Not the parasite don't allow those cells to become pro-inflammatory mature cells, but keep them as semi-mature. Those kind of dendritic cells we called tolerogenic cells, and they promote the shift from pro-inflammatory response to anti-inflammatory one that significantly delayed the onset and alleviated and shortened the course of the disease. Next slide, please. Okay, this is just to be very short. On the left side, you can see influence in animal model. Here is what we obtained in a human model of dendritic cells. So in animal model, you can see what is semi-mature status of the cells and which molecules are expressed on the surface. Dendritic cells, have a key role in initiation of immune response during the infection. But under the influence of parasite, tolerogenic dendritic cells became central players in the maintenance of immune tolerance and favorable candidates for cell therapy of autoimmune disease. And right now, there are clinical trials that are going on in Europe which apply those tolerogenic dendritic cells produced under the influence of other agents, not parasitic in origin, in treatment of patients with multiple sclerosis. What we have shown is that excretory secretory products of Trichinella induce stable human tolerogenic dendritic cells at that dose. Induction of those cells involves the engagement of multiple pattern recognition receptors that you can see on this slide. We call them toll-like receptor 2, 4, then dendritic cell specific ICAM-free grabbing non-integrin sign molecule and mannose receptor. Next slide, please. Until I can see next slide, just to mention that this kind of cell influence that shift to Th2 and T regulatory cells. Next slide, yes. This is just showing what kind of pathways are going downstream in the cell upon interaction of parasitic antigens with toll-like receptors, and we showed that there is transient activation of ERK and mild activation of P38, meaning that on this way parasite will stop production of pro-inflammatory cytokines from those cells. Next slide, please. In addition to reduce pro-inflammatory phenotype and function, tolerogenic dendritic cells express some tolerogenic markers such as IDO1 and ILT3, and those cells enable that T regulatory cells are doing that way due to IDO1 dependent manner. Next slide, please. Those are the results that we obtained by FACS system, meaning that we use that fluorescent method in analysis. And here you can see that T cell activated with excretory-secretory product treated dendritic cells, educated dendritic cells, exhibit suppressive feature. Those T cells inhibit the proliferation of peripheral blood mononuclear cells, and functional properties of educated dendritic cells are preserved even in inflammatory environment. This suppressive activity could be ascribed to increased percentage of T regulatory cells that appeared after the influence of trichinella educated dendritic cells. Next slide, please. So when we proved that infection can dampen or prevent EAE, we found protective effect of infection could be established by not by a live infection agent, but also by different ways, other ways to be successfully treated animals with educated dendritic cells. And on that way, we also ameliorated the disease. Also, the prophylactic application of excretory-secretory products or its components by itself exerted beneficial effect on outcome of EAE. Next slide, please. So here is the example how everything looks like when we applied educated dendritic cells. You can see that there is a lower level of maximal clinical score, cumulative index, that we shortened duration of the illness. And when we compare the achieved effect by dendritic cells with the effect of infection, we can see this almost the same result, that these stimulated dendritic cells maintain switch towards to Th2, meaning IL-4 presence, and the regulatory cytokine IL-10, while there is decrease of the levels of interferon gamma and IL-17, which are pro-inflammatory cytokines. Next slide, please. This is the picture how we investigated the presence of regulatory FOXP3 cells, and you can see that percentage of those cells is significantly increased in the spleen as well as in population of spinal cord infiltrating cells of rats treated with those dendritic cells educated. Next slide, please. Our group was first one who found that trichinella spiralis, like many other nematodes, releases extracellular vesicles as biological nanostructures of ESL1 products, for which we proved to possess immunomodulatory properties, and this shed new light on the mechanisms by which trichinella spiralis deliver information to the host immune system, since extracellular vesicles or exosomes here carry all types of biomolecules, proteins, lipids, RNA, fragments of DNA, metabolites, small molecules, and represent the third fundamental mode of intercellular communication, along with direct cellular contact and exchange of soluble markers. Next slide, please. You can see here the exosomes from trichinella. Next slide, please. And those are the effects we follow. They exhibited the application of extracellular vesicles, exhibit the similar effects on monocyte dendritic derived cells. They induce in completely mature dendritic cells with tolerogenic properties, which remain stable in inflammatory environment. Next slide, please. And those cells exhibited limited allostimulatory capacity on T cell responders, and as I mentioned, remain stable upon introduction of inflammatory stimulus. What we wanted now to do at the end, we wanted to find the most favorable, if I can say, approach how to treat humans, not to use infusion of educated dendritic cells, but to find a way how we can easily apply components of trichinella in curing the disease. So we designed the delivery system in the form of implant consisted of biodegradable polylacticoglycolic acid, PLGA nanofibers, and ESL1 antigens. And so we performed implantation 21 days before the EAE induction, and you can see that we achieved very similar results in taming and ameliorating the disease. And now we are showing again what is going on the systemic level in spleen cells. I'm here pointing out on the same cytokines like you had opportunity to see. This is what is going on in the target tissue and spinal cord. So lower level of infiltrating cells, presence of T regulatory and macrophages is increased. Next slide, please. Those are here we have production of those anti-inflammatory cytokines IL-10 and IL-4 again. And the origin of that IL-10 is coming not only from CD4 plus T cells, but also from regulatory T cells, regulatory B cells, and macrophages possibly alternatively activated. Next slide, please. So it looks obvious that besides the beneficial effect of helminth administration in animal model of autoimmune disease and the biggest results from few clinical trials which use the live worm as a therapy, this kind of therapy faces a lot of obstacles and carries risk for the host organism. From that reason, we need to learn more from trichinella as master of immunomodulation about component its uses and mechanisms it utilizes, striving to contribute novel therapeutic approach, not only to multiple sclerosis, but also other autoimmune diseases. So next slide, please. I like to acknowledge the work of members of my group, some national institutions that are involved in, as well as collaboration with institutes from France, from Japan, from Italy, from United States and United Kingdom. Thank you very much for your attention.
Many thanks, Liliana, for your excellent presentation with a lot of facts and immunological points. Are there any questions? We have time for one or two questions. A question by Isabelle Vallée.
Okay.
Hello, Liliana. Nice to see you. And of course, we would prefer to have you in presence here. But thanks for your presentation. Just, well, general question, but within your ES antigen, do you think that Tivlos is the major antigen that is involved in what you observed, or do you think about a particular protein within the ES antigen?
Hello, Isabel. I hope that soon I will feel better enough to meet you somewhere in Paris, and I'm also missing my presence among you over there on place. That is a nice and interesting question, because as an immunologist and knowing well that excretory secretory products are consisted of many glycoproteins, and in that glycosylate part that the Tyvelosis is specific and characteristic for trichinella, I thought that it will be very hard task to produce a recombinant antigen with not only consisting of protein part of the molecule, but also to consist Tyvelos, and that only on that way we can imitate what parasite is doing. But I think that Tyvelos has important role, since we, although we were able to show that parasite interact with those glycoparts with the receptors on the surface of the cells. But I had the opportunity to read a paper from our colleagues in Japan that I mentioned before, who succeeded to just apply recombinant protein from Trichinella pseudospiralis, not glycosylated at all, and to tame and to cure somehow in animal model signs of autoimmune disease, which name is psoriasis. It is a skin disease, or also autoimmune disease. So the question is open because we really don't know how important Tyvalos is in that tolerogenic process induction. Probably time will show that. I don't have a real response on your question.
Thank you, Liliana. Thanks a lot.
Many thanks for your presentation.